and she will introduce herself, and this is the first of the 3 of the series that's about breeding programs. Thank you.
Hi, this is I'm Sue Cobey. I work half-time at Washington State University, and I run a instrumental insemination business, training, things like that. So what I want to give you today is more of a background, basic biology about bee breeding, because I think a lot of this stuff it's well known. It's a basic review, but there's a lot of things in here that I think are necessary to take another look at. Mostly because honeybees are so complex. I mean, of all the animals you breed, I think honeybees are probably one of the most complex. So I want to talk about that complexity and then kind of bring it home and give you also the very simplicity of how to run a breeding program long term, which is really what this is all about. There's been a lot more focus on pests and pathogens, obviously, because the things we're dealing with, malnutrition, chemical residues. So everything's gotten even more complex than that. But I'm just going to give you a good basic overview of some of the challenges, some of the things that we need to really look at. Yeah, okay, trying to get my, okay. One of the basic fundamental pieces of information you need is to look at sex determination, honeybees. It's more like plant breeding than animal breeding, because the drone is haploid, which means it's got one set of chromosomes. It's from an unfertilized egg versus the queen, and workers that are diploid would have 2 sets. So there's one gene that determines sex, and there's different forms of this. You may have 6 to 19 different alleles. Alleles are different forms of this gene. And if you have the same allele with the sperm and the egg meet, you get a diploid drone, which is not viable. The bees will, as soon as this hatches, the bees will remove it. You can rear these in the lab. I think Voike did this many years ago, but they're semi-sterile. Not sterile, but they have small sperm that you can do that in the lab. The other thing is if you have the more alleles you have in your population in this sex gene, the better brood pattern you're going to get. Basically, it comes down to that. If you have a lot of drones with similar alleles, you're going to get kind of a spotty brood pattern. So that's a really basic concept that you need to know. Honeybees are really highly sensitive to inbreeding, and this is why. Drones. We have an incredibly high mating frequency. Drones, think of them as kind of flying gametes. All the sperm in one drone is identical. So for the queen, she wants to mate with as much diversity as she can. That's why you have this really high frequency of mating. She mates in flight with many drones. That's very risky, but it also brings that bit of genetic diversity which you want in the colony. That's really important for her. This whole process is very random, so it's difficult to control. This is one of the main things about breeding honeybees. It's really difficult to control unless you have an isolated mating station or use instrumental insemination, something on that level. So every time that select queen goes out to mate, you get this kind of randomized mating, and you kind of go back to mediocrity. So it's really important to get control of the mating of the queen. Drone congregating areas, we know that the drone, you'll get drones from 10, 30,000 different combinations of drones from, it could be 200, 300 colonies. So you're looking at a really genetic mix in this gene pool of what those queens can mate with. There is a sort of mating, which what I mean by that is we know that Carniolans will mate at a higher elevation versus Italian queens. So you'll get a little bit of that mixing that way, or sorting out that way. But these areas are very specific. It's like a clearing among trees or between mountains, like a valley where they congregate, and it's really stable over time where they meet like this. The virgins tend to fly kind of low and out. They fly further than the drones. They're looking for that, again, I think they're looking for that diversity, that challenge. They don't want to mate with drones from their own colony, so they're flying at a further distance. And the semen storage, this is crazy, because when that queen mates, she's mating with what, 10, 20 different drones. But of all those drones she's mated with, 90% of that is expelled. But the interesting thing is that she retains semen from all the drones that she mated with. She'll take just a little bit. For example, one drone produces about 10 million sperm, yet the queen will only store about 5 to 7 million sperm on average. That seems really inefficient. Half a drone's worth of semen is what she stores in the spermatheca after collecting that massive amount of drone semen over many 2 or 3 flights. So it seems very inefficient, but it's efficient in terms of maintaining that diversity, genetic diversity. And what you're looking at here is, this is a virgin queen, the spermatheca of a virgin queen. It's crystal clear, it looks like a diamond, it's quite pretty. And this is the spermatheca of a mated queen. It's kind of a cafe au lait, creamy, same color as semen. And during the mating, you can see here these oviducts expand, and the sperm, it takes about 40 hours for the sperm to migrate up into the spermatheca. So what you see is, I don't know if you ever notice those little ribbons after the queen mates or something, but she's discarding a lot of that semen, and the worker bees will help her remove that excess. So this is an amazing organ, the spermatheca. It's got a net of trachea around it. It supplies oxygen, and it also has lots of nutrients supplied to the spermatheca. How that queen can keep sperm alive for years is really a mystery we'd like to know. So this is what you're looking at here is a spermatheca. The sperm is got really long tails, and you would think that this would just bundle and mix and be difficult, but we've seen that. We thought this was like tightly packed, and it would be like drones would be used over time individually, but we know now that the sperm is fairly well mixed. There's a little bit of clumping in there, and you'll get different amounts of sperm per drone depending on the drone. Maybe something in the mating sequence there, I don't know. But this is what I'm going to show you here is this is the spermatheca. This is what those swirly patches you see, that's the sperm moving inside the spermatheca. So this stuff is mixing, always constantly mixing, which is a pretty amazing thing to look at. Okay, drone. The drones in the breeding program, this is the most difficult part, I think, to have mature drones from the select line you want, when you want them, in the numbers you want them. This is really your most difficult job, I think. During the season, peak season, swarm season, lots of drones available, it seems easy, but the drones are the most perishable. If you have any stressors, any kind of issues with pests, pathogens, chemical residues, you can have nice looking drones, but they may not have good viable sperm. Dave Tarpey showed that there's a lot of differences in the viability of the sperm that they have. So, and if you look at a colony, probably about half of those drones are going to be immature. So, and the most mature drones, if there's a stressor, those are the ones that are going to get kicked out. So you want to take a very good care of your drones. Big demand on nutrition for drones. They need lots of pollen for that sperm production. So take care of these boys, really important. Recombination. This is another amazing factor about honeybees. They have the highest level of recombination of any animal. That's like 20 times more than humans. What I mean by recombination is when the egg and sperm meet, the parts of the genes, they do a little dance and they change parts and pieces. So you end up with the individuals are different from their parents. You get these new combinations. This is why bees are so adaptable to different situations, climates, something new in the environment, a new pest, disease, something like that. It's a way to test how to take advantage of these, how to adapt, change. You can select very hard on drones because it's kind of a clone, but at the same time, all those different worker bees, all those different, you have those different subfamilies represented by the different drones they mated with, and then they're going to do this recombination. So you get a lot of diversity within that colony. We call this a superorganism, the honeybee colony, because it's so complex. Each drone that queen mated with, you have this subfamily representing that genetic component, but you have many subfamilies in a colony, and they may have different traits, different propensities, different levels of how they react to behavior, things like that. But there's no central rule, no central control in that colony. Think of that. You've got this really complex social unit, but there's no major control. It's all about stimulus response. It's individual bees picking up cues from what they acquire within the colony. For example, if you have a really hot spell, then all of a sudden the need for water is going to be really intense. So they will do major water collection. Or if you've got a lot of brood in there and the larvae put out a pheromone, hungry, feed me. So you get a lot more pollen foragers coming in, things like this. So it's a very dynamic, flexible, moving thing. No individual bee has an idea of that whole complexity. It just knows that it's responding to whatever that stimulus is at the time. Does that make sense? Just that complexity. So, and these individuals will have different levels of threshold levels. For example, there's sensitivity to something. For example, like the water collection, you may have some that have a low threshold, they're very sensitive to that, or pollen, where the more diversity you have within those thresholds, the more average, the more stable that colony is. Where if you select real heavily in one trait, you'll skew that population, and sometimes they don't respond with as much stability as if you have a really lot of diversity within the colony. And again, things like pheromone communication is really important too. The queen and the pheromone, the cohesion in the colony. And this does regulate different colony conditions, different demands, needs, whatever the colony is needing at the time. Stimulus response is, again, these threshold levels, they vary. And it's all about the group dynamics, how they fit together. This can change, it can alter behavior. The stimulus can become more and it can recruit more bees. For example, if you have a colony with a small percentage of Africanized bees, that alarm pheromone will recruit the European component of that hive, and you'll get a more defensive response in general. It'll be more African-like, but maybe the colony's heavily European, just has a small subset of African bees. So things like that. It's all about colony needs and being dynamic and flexible and resilient. We see this with thermoregulation, wintertime, hot summer, how they adapt to that. You see that in the winter, they get a tight cluster, things like that. Defensive behavior, we talked about that a little bit. Resistant suppressant diseases, if you got a new pest parasite of some kind, you may have one subfamily that shows more resistance, just enough to give it more survivability, versus if they're all on the same level and they're more all susceptible, they're gonna perish. So all those little things add up. Foraging, if you've got more different threshold levels for food, pollen, nectar, pollen, whatever that is, they're exploring different resources. So the more diversity you have in the colony, the more likely they are to find new sources and bring that in. Natural artificial selection. Selection is just what you're trying to do is take those heritable traits and change the frequency of those over generations. want better wintering, more honey production, better hygienic behavior, all these things. So you have the genotype, which is basically the potential. It's the genetic makeup of those bees, the diversity within the colony, it's the potential. The phenotype is the expression, what you see, the behavior you see, and that's based on the environment they're in, the past experience in their network of social behavior that can change behavior, things like that. So our goal as bee breeders is we do artificial selection. And that really depends on how you can do that, fast or slow, depends on that population size. The bigger the population, the slower your selection is going to be. The strength of your selection, what I mean by that, how tightly you do that selection process. For example, in my program, first working with Marla Spivak and hygienic behavior when this goes way back, early 90s, I guess, but I wasn't willing to eliminate some of my best performers because they weren't hygienic. So you keep those in the gene pool. There may be lots of other traits contributing to that, I don't know. But so it's the level of how you select like that. So, and when you do that selection, there is a loss of genetic diversity. You're selecting out what you want in that population. So you're going to eliminate some of that diversity. It's just normal to do. That happens in a natural population. You talk about genetic drift, just pressures from the environment will produce something, change that over time. The analogy I really like is Rob Page's balloon. He uses this a lot, where you take a balloon. I don't know if you guys have ever made those little balloon animals. You push something here and something else pops out in a different place that you didn't expect. That's really what these traits are like. They're polygenic, which means you've got a lot of different genes working together, influencing each other in this network. And some of those genes are pleiotropic. What I mean by that is one gene can actually act differently, have different effects, sensory systems, behaviors, things like that. So if you look at this whole picture of that colony, it's dynamic, it's flexible, it's constantly changing. You have a lot of diversity in there, a lot of influences from the environment playing on this. So that makes your job really hard to do selection for the specific things you want. You can, I like to look in terms of like a population. You want to slowly move this population towards the desired traits that you want over time. And that's basically keeping selection pressure on the population over time. So if you go too far in one direction, maybe you'll get something that pops out differently. I don't know if you've read this book, The Red Queen. It's Lewis Carroll's take on Through the Looking Glass. He's got Alice running, and she's constantly running in place and making no progress. That's kind of what you look at in these pest parasites, where the pests are developing more resistance to the controls that the bees develop, or the chemical pesticides we use to control them, things like that. So it's a constant race off and on. And if you look at that, the parasites diseases often have a faster development level versus the host reproduction. So it seems like they would always win, right? They have the advantage here, but it doesn't make sense to kill your host. So if you look at the bees, that high level of genetic diversity and the high level of recombination, which is an ability to test for new traits, things like that, really gives the bees an advantage over all these different factors. We know that bees with a high rate of polyandry, what I mean is that poly means many, drones, mating with many drones. You have this more diversity within the colony that increases. There's lots of studies showing that the more diversity you have within the colony, the better overall fitness you have, more productive, higher population, more resistant, less prevalence to pests and diseases, better survival, etc. They just manage better because you have more stable colony, more ability to change, react to different things in different ways. So a lot of work is going on with like single trait selection, mostly looking at, I get this question all the time, well, there's one gene for one trait. No, there's not really one gene for one trait. A lot of these genes affect several traits, and most traits are polygenic, which means they're made up of several different interacting traits, and those are all, again, flexible and dynamic. You have these networks of interacting genes that affect a lot of different traits. They change the behavior, physiology, even the anatomy of the bee. For example, if you take a look at Rob Page's pollen hoarding bees, those pollen hoarding bees have more ovarials. I'm talking about the worker bees. They will turn into laying workers faster. They have a later date for foraging. They have different levels of vitellogenin. This is a precursor to egg making, and juvenile hormone levels, things like that. So it's actually changing their whole physiology, not just the behavior. And that's kind of amazing. So when you do single trait selection, you need to look at those other factors in there, which may not be real obvious when you first start doing that thing, because all these things are correlated. Bees are they call it plasticity. This means they're just the adaptability, the resilience, the ability to adapt to whatever's going on, but then come back to that resilience when whatever that stimulus calms down again. So it's all about the dynamics of those things. Hygienic behavior. This is a genetic specialist within the hive. It's a very high value trait, obviously. It's high olfactory sensitivity. The bees have a really good sense of smell. So they're detecting and removing, sometimes recapping the cells and trying to get rid of diseases, preventing diseases. It's a recessive trait, which means it needs to be homozygous, which you have to have it in both the drone and the queen for that to be expressed. Heterozygous is if they're different. Homozygous means they're the same. They both have to carry those genes to have it expressed. And it's a common trait, but it occurs in a low frequency in the general population. You can find it in any population, but unless you select upon that, you may not get a high frequency expression of it. So that gives you value there. But then again, you have to it's kind of a trade-off. How heavily do you select upon this particular trait? There's a cost to that. I don't know if you remember the early super hygienic queens that came out. I got some of those, and they would chew down to the midrib. They would cannibalize the brood. They were just so extreme. But if you took that and put it in a diseased colony, they would just clean up everything and it would be fine. It was great. I used to take a frame of emerging brood from those super hygienic colonies, put it into another colony, and that would be really nice. So you have to kind of look at the level of that, and you want to make sure you're not excluding some really valuable traits that are important to the production of that colony. So those things are important. Again, that high diversity within a colony, it's makes everything kind of average. You don't have these extremes in whatever it is, hygienic behavior or thermoregulation, or whatever the trait is. It's very stable, which means oftentimes those survive better, and you do have more fitness in that diversity. So that's kind of what it is. Hey, we live in a toxic world. This has to be considered, all these different stressors. And if you're given these stressors a little bit one by one, the bees can handle it. But this multi stressors, they're synergistic, they act on each other, and it's just too much. So you see, if you have malnutrition, the immune system breaks down, you're more susceptible to pesticides, pathogens. You have a shortened lifespan, reduced foraging activity, the colony population goes down and it collapses. And that's kind of what we're facing today. So you really have to kind of look at all these factors. But at the same time, exposure to those different things gives the bees the ability to develop resistance on a certain level. I mean, if they're not overwhelmed, they'll deal with it. I think tracheomites are a perfect example. When those were first introduced, they were pretty devastating, and then they kind of fizzled out. They're probably looking at all the miticides we put in colonies. Maybe they don't have a chance right now, but anyway, but these, what you have in exposure to those things is over generations, you have this immune imprinting system. The bees will learn this and they'll pass it on to the next generation. It's epigenetics. So you want some exposure on a level the bees can handle that, adapt to it, and develop that. Bees that can, I'd like to get back to where we have bees that can live on their own, develop on their own. It seems like now we have to take care of them almost too much. But okay, so I've been thinking about this a lot. I'm stepping out of my New World Carniolan closed population just because it's time, and kind of looking back at this whole thing. I've gone through 40 generations with this, which is impressive. But the key to the longevity of this program is the simplicity of it. And that may be initially being young and a bit naive going into that program, but I had some really good teachers. Page and Laidlaw were developing their closed population theory at UC Davis when we were there. And their concept was to work with a population and just select the top performers. And that's something I think beekeepers have done always. But the difference is to control it with instrumental insemination and strengthen it with a good selection program. And that's the only difference, really, what I've done with this. And again, it's just looking at when tracheomites hit and varroa hit and all these different things hit. What Paige suggested is you just go for the best and you'll bring those traits in. We still don't know what is resistance to varroa, but if you just pick out the ones that are doing a little bit better than everybody else or maintaining lower levels, that's going to give you more ability to move the population in that direction. Early on in the program, I did get some VSH stock, and because the California guys wanted me to add this to the New World Carniolan program. And when I sent those queens down, I inseminated my virgins with the semen from that. They said they were less productive, and they also had higher levels. I don't know what that was about. I really don't know what that was about. I'm not saying that this population was more resistant, but it's just that I think it's more diverse, better fitness, better able to deal with those things or something. I don't know. And wrote a couple articles about this, applying this, modified it a bit. If Page and Laidlaw took it, it's a theoretical model, computer model. You can go 20 generations. So we wanted to look down the road. How do we keep this going? How can we enhance it more? And that was basically by opening up the population in terms of looking around. Whoever had Carniolans, I would always look, I would set up a separate yard. And if what I liked, I'd take a handful of drones and throw that into my gene pool. So you don't have a big change in there, but you have more, you're bringing in more genetic diversity. It's pretty simple. Basically how Page and Laidlaw said, if you start with 50 different queen mothers, you can go 20 generations. So basically we collected stock from all over the US and some from Canada. Back cross this, try to get more Carniolan-like characteristics. And then from those, you take virgins off of those, and you take the pool of drones, take semen from the pool of drones from all those, inseminate those queens and make a test population of a couple hundred colonies. Those numbers varied over the year depending on what was going on. But then from those, you take about the top third of those. These are your breeders for the next generation. So it is a type of inbred system, but it's a big enough gene pool that you don't have effects of inbreeding. And then we would just reproduce this cycle every year. So you got your 50 queens, mothers, you're taking virgins and drones off of those, inseminating those, crossing those, make a test population, and then run them through the selection process. And it was pretty simple. We're looking at brood viability. We're looking at buildup coming out of winter, any incidence of diseases. We'll do a weight gain test for honey production. Pretty simple, straightforward, general characteristics. And you just slowly skew that population. Sometimes you get things maybe you shouldn't have put in there or selected, but it kind of comes out in the wash. And you're not dependent on any particular queen. The programs I grew up with was the Starlight Midnight 4-way hybrid. You got these 4 inbred lines, cross them, and that's your end product. And that was really cumbersome and labor intensive and risky because those inbred lines are really hard to maintain. So the concept of working with a population like that, it just made sense. So we went with that. This is kind of a little history. We started this in the early 80s in Northern California. For about 10 years, we ran back in the 80s just producing Carniolan production queens. And then I think the big event that kind of shut us down was the border closure to Canada. That was a little bit painful. But then but it was a good opportunity at the same time. I moved the program to concentrate more on the breeding stock. So that's really the next 10 years of that. And then as I approached that 20-year wall that was predicted for having some inbreeding and things like that, we started looking, I was partnering with Steve Shepard, and we started looking at, can we import Carnica semen from abroad? We tried this in the early 80s, and just the politics, what was going on, it wasn't an option. But over time, especially when they started bringing in bees from packaged bees from Australia for the almond pollination, that kind of opened up the door. So that was kind of the next step. We brought in some semen from the Austria Carnica Association, the German Carnica Association, and Slovenia. Slovenia is an amazing country. The whole country is Carnica. And you see, it's very agriculture, very rural, and these everybody has a beehive. Every little town has a shop selling honey and bee products. They're very proud of their bees, and they still run these bee houses. I don't know if I have a picture of the inside here, but the frames just pull out. So we went there, we collected semen. This is my little tube of semen here. And our permit was restricted to semen only, as it should be. This is a breeder, Stan Put. He's this is way up high in the mountains, and they move the bees up here for the queen production and honey production. And here's a little house here. It's got a place to sleep, and you can cook a meal. And these are some of the drone mothers up here. And these are the mating nooks. They have to cage these because there's a resident bear. So just to keep them safe. This is what they use as a smoker. It's just a piece of pressed board that they light. That's the extent of a smoker. And this is the most beekeeping equipment I wore. I never put my veil on in that country. These bees were just so really elegant, beautiful, easy to work with. Really nice to work with them. Partnering with the California queen producers. This is really a key for us, especially once I did only the breeding stock. I was really dependent on the commercial queen producers to mass produce these queens, get them out there to the industry. So that was a really important part of the process. So now I'm transitioning this to Strachan apiaries and Buzz's bees, Valerie Strachan and Buzz Landon. this is pretty new. We just did this last year. So I will still go down there and help them with the inseminations. And also we have on these collection trips, we also cryopreserved semen, which means we put it in liquid nitrogen. This is a little experimental. The queens there is some damage there. The queens are not that productive, but it's enough to recover the stock. So we'll continue to augment the stock with that, and I'll help them with the whole process here, and then hopefully they'll take over this and let it go from there, it's pretty exciting. It's just wanting to take that into the future, basically. So instrumental insemination, it is a bit technical. It's not so much difficult, it's just precision. If you're interested in this, it takes a bit to learn it. It's tedious, precise. A lot of it is handling virgins and drones. Most beekeepers don't like to do that. Even among the big commercial queen producers I work with, working with virgin queens, they want cells or mated queens. Drones are just so perishable, and you really have to train yourself how to deal with that, how to work with that. The insemination, once you get that down, it goes pretty quickly. And this is really, I put this little diagram in here just to show you. So if your first generation, you got this beautiful 100% queen that you like. So her daughters, that first outcross, you're going to lose 50% of that in the first outcross of those virgins, unless you have stacked the mate or saturated the mating area she's in. So this goes down pretty quickly. So I think this is one reason that breeding programs have been so difficult, because of that open mating, that just random open mating of all those drones in the drone congregating area makes it really difficult. So I encourage all of you to, maybe in Canada, you have more options for isolated mating stations up there. I hope you do. If you do, that's much, much easier, obviously. But there's been a lot of new interest in insemination. And I've had people call up and say, well, I can't get my drones together. I'm going to inseminate these instead. Well, that's really not an option. You need to get the drones together. That's the hardest part of this whole process, I think. I put this is an old graph I did, mostly because I got tired of people telling me that these queens don't perform. Instrumentally inseminated queens actually can last longer than a natural mated queen. And I say that because you can give them a known amount of semen, and usually you're putting all the optimal, you've got selection there, you put all your efforts into the best raising of queens, things like that. So you can, I've had these things live 3-4 years, and they can do that. So this is, if you go to my website, you can read this paper. It's got a lot of information about care of queens and why all these things are, but I'm just going to briefly go through this chart to give you an idea. So these are all these different studies across the world. They go back to the mid 40s to the present. I probably should update this. And the blue is natural mated queens. The yellow is instrumentally inseminated queens. These are the number of queens they compared. This is honey production between these two queens. Some of these are in kilograms. I think I covered all the kilograms. Okay, brood, amount of brood. This is centimeters squared, eggs per day, number of cells, different figures there, but they're comparing across these two. And then you're looking at, then you're looking at how the longevity of these queens, how long these queens lasted. Most average is about 1.5 years, 2 years. And then you compare the treatment of those queens. And a lot of these queens were, let me move this over so you can see it. You're looking at the introduction methods. So, okay, first I'm going to group one is equal performance between the II and the natural mated queens. Group 2 is high performance of inseminated queens, and that's really based on selection. And then the study down here by Harbo is the natural mated queens had a higher performance. And the big difference between that, so if I lump all these studies and look at how these queens were treated in terms of the age the queens were inseminated, the amount of semen they were given, and the introduction methods. Okay, these queens were between 5 and 10 days, which is that window of time when a queen naturally mates. You know, if you get really bad weather, after 2-3 weeks, those queens don't mate well. So these queens were, in Harbo's study, these were banked for 2 or 3 weeks before insemination, so they're not in their prime. They were given 2 doses of less than 3 microliters, where all these queens were given 8 to 12 microliters. I generally tend to give them 10 microliters now. You get about 1 microliter per drone. So they were given bigger semen loads. And we know that how you introduce those queens, if you look at a virgin after, or a newly mated queen, she's very active. She's running around, the bees are feeding her, helping her remove the drone parts. And that activity helps sperm migration versus if she's banked by herself, isolated, she doesn't have that movement, which is really important for the sperm migration. So what Harbo did was he banked these queens another 2 to 3 weeks. So these queens were a month to 6 weeks old before they ever saw comb. That's the difference in this study between all these queens and that. So if you're going to do this, make sure that you pay attention to the care of those queens, because that will really influence your performance of these queens. So I didn't get, let's see here. Yeah, I didn't get too much into the whole selection process. There's a video here you can watch. We did a video that talks about the selection methods. But again, in my mind, it's keeping it very simple. That whole selection process is just, you have the queens are all at the same population level, the same age. You just run through these, and it's a comparison test. How solid is the brood pattern? Are the bees flighty? Are they very gentle? You're looking for population. Is this 4 frames of brood, 6 frames of brood? You're just going through there. Is there any evidence of disease? And you do that a couple times over the season. And as they build up, we did a weight gain test looking at productivity, and it's pretty simple. And it costs you 5-10 minutes a colony over the season. So, and the more colonies you look at, the tighter your selection can be. So it's really that, and it's really based on looking at a population, skewing that population, maintaining high diversity, genetic diversity. There's just many studies showing that the more diversity you have within your population, the better fitness you're going to have in general. And all these different traits, all these complexities, we don't really know what makes Varroa resistance. And you've got something that jumps species. It's complicated. It's very complicated. So it's going to take a lot to get to that level, but we can give them more ability, selecting for hygienic behavior, selecting for grooming behavior, these things that we know are additive benefits to the bees. We can do that, but it's really fitness and maintaining a big population in there and skewing that toward productivity. Anyway, that's my take on bee breeding. So, you're having a symposium in July. I'm not sure how this is going to go. It might be right now, I think it's going to be a hybrid. It might even be just all online, depending on the COVID and situation here. We'll see. But this will be focused on insemination and also cryopreservation. This is a really interesting thing that I think in Europe, it's more of a concern because there's a need to conserve different subspecies, ecotypes. There's 28 subspecies of honeybees in the world. We use 3 or 4 commercially, mostly Italian Carniolans in the US. And there's a huge resource out there. So And all these are under a lot of threat with pests and diseases, lack of forage, moving things around, integration of other subspecies in there. So there's a big push to conserve these. And also, if you look at this whole process, you can just put your whole breeding program in that tank and then bring it out, give it to your grandkids down the road. That's how amazing this is. The technology is developing. It's a little, as I mentioned, we have the same success you have with like cattle or horses or chickens. The difference is those animals only need a few sperm. Honeybees, she needs 5 or 6 million sperm that's viable. So we're not quite there yet, but it's enough to recover the stock. And there's a lot of people working on this worldwide. So I think we'll get there eventually. Something else that's interesting is that the USDA has now put honeybees in their genetic repository. We've never had that before, different animals, plants, but honeybees, we didn't have the technology. This is Brandon Hopkins in the picture here. He just tweaked this enough to make it a little bit more practical. So that's mostly what this symposium is going to be about. So thank you. And I don't know if we have time for questions. Let me see if I can pull up my chat here.
Hi, Susan. Thank you so much for your presentation. It's been really super informative. I definitely have a question. I wonder, this is Emily Huckster. I wonder, it seems as though queen longevity has reduced over time. And I don't know if you agree with that statement or not, but do you think it's partially because we maybe don't wait long enough before we start breeding from queens? We're not letting these, we're not waiting 3 years before we pick a queen as a breeder. And so we're not finding that longevity in our stock anymore.
Yeah, that's interesting take on that. Yeah, I'm sure that could be a factor, but I think more importantly, it's got to be I noticed since Varroa, the quality of the queens is less. It's really hard to get rear a high quality queen, nutritionally, I think, because of all the parasites, pests. You got viruses, even if you clean up the Varroa, you still have viruses, and malnutrition, pathogen loads. You've got pesticide residues. You've got this underlying kind of toxic environment we live in now. So I think it's attributed more to the combination of those different things. Hey, and congratulations taking over your mom's outfit.
Thanks. Trying.
Yeah, I've fired her a couple of times in the past.
Yep. No, I know that one. It took me about 5 years to try to, yeah, it's hard. It's hard to let go, give it up. Anyway, but no, I think it's just so many different factors in there. But that's interesting what you said, though, that you're not looking at queen longevity as much as we used to. I know people that bought my breeders, they just get new ones every year and kill off the old ones. And then I had one of them focused on the older queens. Some of these were 2 or 3 years old. And I know I've done that too. Just an old queen, and she gets lost in the shuffle and she ends up in the production level and then comes back around or something. But I've really begun to treasure those old queens. So yeah, keep them around. There could be something to that. I don't know. I haven't seen any data, but there has to be something to that. Maybe she just got more sperm. Maybe she was just reared in a healthier environment, less exposure to pathogens, chemicals. I don't know what the deal is, but they're special.
We do have a number of questions here as well.
Okay.
And we have another few minutes if you're willing to help us out with a few answers.
Yeah, sure.
Where do you see the genomic markers fitting into a breeding program these days?
Good question. No, that's an amazing ability we have. We've learned so much from that. But again, keep in mind that it's not straightforward because of because that recombination, that complexity in there, you can identify them, but there's all these things that are interacting with them. So it's really hard to, okay, say you found this marker for this particular trait. It's really hard to maintain that in the population because
And I
it's
think-
change in mass and environment and the complexities of a hive, recombination, all those things play. But it's going to be a lot more difficult with honeybees versus other animals. But what an amazing thing to have.
Right. I think as well, like reading Rob Page's book, The Spirit of the Hive.
Yes.
I think that really gives you an idea of the complexity, but also it made me want to question as well when we have these breeding programs, when there is this syndrome, as he calls it, for the pollen collectors, and it looks like you could dump into that as well VSH and hygienic behavior, just because of all the same traits that you see when you look at those, like sensitivity to light, learning heightened, higher activities, higher pollen hoarding, and all those things that kind of fit into a package, more or less.
Yep. Yep.
And then if we have this breeding program that's kind of looking for gentle bees, which and honey in particular, which is kind of the opposite to this pollen syndrome type of a bee.
Yeah.
How
honey,
are we
honey's
doing?
so complicated. That's such a complicated trait to select for.
Yeah. So I mean, I would say, and I'm to give a talk in the next couple of weeks, and I'm going to say that honey production, just let that happen. It will happen. Go ahead and breed for everything else. So is that valid in your mind or not?
I think so. Yeah. No, I mean, you still, according to Page, he said, just select the top performers and you bring all those things in. You don't have to identify all those traits. You just bring that into your gene pool. So he has another book out, Art of the Bee. Have you seen that?
No.
Oh, get that one.
Okay.
And rumor has it that he's redoing the Page Laidlaw coloring book.
Ah,
And I'm sure that will be quite different, I'm guessing.
but that'll be fun too.
No, if anybody out there has not read Rob Page's books, hey, I'm the biggest fan, but he just takes his whole career and he brings in all these other research things to explain things, and it's amazing. And if you read it again, because there's so much in there.
Yeah, that's so true.
Yeah.
Wendy, what other questions? Wendy Gilson's also helping out with this question and answer period. Do you want to bring out a different question, Wendy?
Yeah, one of the questions was, do you think a national breeding program is something our industry should strive towards? And I would go on to also ask, like, if you think even a provincial or regional or neighborhood breeding.
Yeah, well, it's if you look in Europe, they have all these different like regional breeding programs working with mostly their native bees or bees adapted to the area. There's a lot of value in that. But if you look at the US, probably Canada too, we're pretty mongrelized, the population. But I think if you could kind of isolate yourself and select out what does well in that area, there's a lot of value in that. But as far as a national, what I'd like to see is a national stock center where you could get you could buy semen or get your queens inseminated or make these different combinations, have all these different lines. That's kind of what the Baton Rouge lab was proposing to do early on, but it never happened. And it's been kind of my dream to set something like that up, but the climate was not right. It might be now, but to fund something like that would be pretty expensive. I think Canada is unique in that you're a smaller, more focused country, and your beekeeping needs are more focused too. You need good wintering bees and that hard, heavy honey production then shut down. So hard to answer, but yeah, no, I think maybe for Canada, a national bee type of thing, or you could have a regional, like a East Coast, West Coast, or something like that.
One other question we have here is, do you number your breeder lines and or for tracking?
I don't really do lines. It's a population. So every queen has a numbered tag, a number and a color. Color is usually the year and then they're numbered. So, and I just do mass selection. I just take the top third of those. Don't really care if how many dollars are represented in there. It's just kind of mass selection. But I pay a lot of attention to brood viability. If I have something that doesn't have a really good brood pattern, then I'll backtrack. But I've never really run into that because I'm taking those top third, I'm taking versions from those, and I'm also taking a pool of drones. So I mix drones up from all those colonies to inseminate with. So it's not really like separate lines. And it makes the record keeping way more easier.
That brings up another question. Do you take your notes in the field on paper, or have you found another method that works better for you?
I'm pretty old fashioned. I'm pretty much out there by myself. So it's paper and then just a simple Excel file. You know, now I would change it. I would do something a little bit fancier that's easier. We have all these electronics and stuff like this, but you know, it's mostly just me out there. So I haven't really converted that over. So another reason for the next generation to take it over.
So we have a couple of questions here on the chat. Let's see. Oh yeah, so you don't use barcodes or anything, okay. Do you note other traits other than the 4 or 5 that are your standard criteria?
Yeah, sure. If you see something in the field, like if you have like a super pollen collector or something, I'll just put a little P+ on there or whatever little trait. There's always little notes in there, and it can be quite messy sometimes with that. Yeah, but the important thing is to give those basic values a point so I can add them up. And really it comes down to if I have something that's on the borderline, and I'll look at the notes and say, oh, but she had this, so then I'll put her in the pool too.
Yeah, that sounds very reasonable.
Sometimes I'll take my, a little larger pool of drones. Like I'll have like a second tier drone pool like that. Maybe they didn't make the cut for grafting, but they're marginal not marginal, but they're still pretty good. So just to keep that diversity, I'll take a little bit more in there, depending on what's going on, how many breeders I got. It's flexible. There's years when I've had many more or many less, or it's just flexible, dynamic, kind of like that. It's just working with a population. You're just trying to shape it.
Another question is, how big is your overall population that you draw from nowadays?
I think the largest it's been is about 300 colonies, and here it's gone down to maybe 100 at times since I've moved up here. That's another reason to give it back to the California guys to expand that again and revamp it a bit. Right now I just have 50 colonies, but I'm not going to produce breeders. I'm just keeping kind of the special stuff to play with, and supply some semen for those guys.
And how would you compare the Caucasians that you've been working with lately to your Carnies?
Ooh, they're, I think my favorite bee is that hybrid. Oh my God. Those things just, some of it may be hybrid vigor, but they are so beautiful, just elegant. You just, I'm totally enamored with the Caucasians. Yeah, but they're more severe. They shut down. I remember the first year I was still being out there trying to collect some drones, and they had just decided we're done with the drones. Bam, finished, all gone. was devastating, but yeah, they're and the propolis, oh my God, they love propolis. So before you get your entrance reducers in there, they've already got it down, and as soon as that weather starts changing a little bit cool, or we get a yellow jacket predation here, they'll start just plugging up everything.
Do you find as well that I see it in some of our bees that for wintering they have a whole different, you know how the classic situation is that bees cluster in the bottom, you've got your honey above, they move up and they eat the honey as they go through the winter. But some of the best wintering bees I've seen actually don't do that. They actually go out and they bring the honey in to right close to their brood nest.
Well, they keep it real tight. I mean, their carnies are like that too, but they keep a really tight brood nest. They do.
Right. Yeah.
Yeah.
With
really
the honey reserves close into them always.
Yeah. Well, they're used to a cold, hard, cold snap. Like we just had that here, and it was like 40s and sunny, a few little things blooming, and all of a sudden we got down into the like 7 degrees. This is Fahrenheit, and I get nervous when that happens because it was so fast. But just going around there popping lids and stuff, they're fine. Probably good for them. Just get that a little. I was out there yesterday. Now it's back up in about 40 degrees, and they're starting to lay eggs already. See, they keep really small populations, but they just have this continuous small amount of brood production that it looks like nothing. And then all of a sudden they just kind of explode population wise. And you wonder how they can do that, but it's just that they just keep that little bit of brood going on when they can. So I just like that production strategy, the way they work. But when you have a really pure Carnica and a pure Caucasian, you can really, if you open the lid, you can see the difference. The Caucasians are really black, just jet black. The hairs on the thorax are very black, and they just look uniformly black, where the Carnies tend to have more browns and grays in the hair. And the drones, really, you can really see that in the drone, especially if you have a colony that's throwing both. They're just really, really dramatic difference there.
There's another question here. As a small scale beekeeper, although I continuously select my best queens as breeders each year, I've been noticing that over time, within the few years, my bees have become more aggressive. Do you think that aggressiveness is due to some Africanized genes mixed in with some of our honeybee stocks?
I doubt that, especially in Canada. I think it's maybe just a touch of inbreeding, or it's hard to say, where that comes from. But it's interesting. The first importation I got, this was the Austrian Carnica Association. Maybe 1 in 20 of those queens, those crosses with mine. My population was nice and gentle, and their population was, but maybe 1 in 20 was very defensive. And what Greg Hunt told me was there's something about when you have 2 very different populations, things happen and change. He said, just take it out another generation. And it did. It disappeared the next generation. But that's a complex trait. I can't answer that question very well. But I doubt if it's African bees. It could be a factor of some inbreeding, too close, too small of a population, or some integration of something else that is mated with those queens. The more different, like especially between subspecies, if you like Italians and Carniolans, the tendency is to have more unpredictability when you cross those, because they're so different, and you get crossover and all kinds of different things going on there. So it's a bit of a wild card. So that's just kind of a guess. I'm not sure, but I don't think it's Africanized.
I have a question. Oh, go ahead.
No, I was just I thought there were no more questions. This is great.
Well, I just was on that note, was thinking that there's defensiveness and there's aggressiveness. And I feel like having a slightly defensive bee is actually, especially these days with the wasp populations, it seems increasing over time, actually a really nice trait
That's
to
Liz's
have.
daughter,
Although
Emily.
sometimes when you open,
Yeah, and the bees will select on that. They will select on that if you have a lot of predation. I mean, if you look at the Africanized bees, is actually more aggressive than African bees in South Africa because they came through that tropical South American, so they became more defensive over time going through that because of bombarded with all kinds of predators and things. So that's just a natural selection process.
and so I guess then again, they would end up in your top group and therefore be selected.
Yeah, if you want that trait, but again, it's trying to keep everything average, really, in that population. You don't want them to get too aggressive, but you don't want them to be so gentle that they're not defending their colony. So it's, yeah. But again, I think of it as like a crowd of people, and they all have a different propensity to react a different way. So you want a few of those wild cards in there. They'll keep the crowd back, but not too aggressive that it's chaos kind of thing.
Right, doesn't keep the beekeeper back.
Yeah, yeah.
Are there any other questions?
There's one more question here for you, Sue. It's what other group or commercial queen producers do you feel have a model breeding program?
What was that again?
What other group or commercial queen producers
Have a model program?
Yeah, similar to yours, but like not yours. Another one or group.
No, if you really look worldwide, there's really few programs that have longevity. Like if you look at the European, a lot of those are modeled after the blip. It's what does that stand for? Animal. It's an animal statistical model that's taken for animals and moved to bees. But it's a whole bunch of heavy statistics. And I wasn't so impressed with the progress. I don't know. There's been a lot of good programs. Like if you take something like VSH, there's been a whole concentration on this particular trait, or Rob Page's pollen hoarding trait. But on the commercial level, it's such a different game because you need productive bees. So, I mean, you've got that Heartland group is trying to do something. You've got Purdue, the Mite Biters group. You got the VSH. The VSH, it's interesting. The VSH thing in Hawaii is changing their whole system over to a closed population, which I think that makes a lot more sense. I don't know. I'm biased. I was kind of weaned on that concept, but I think, I mean, if you look at breeding programs, all different kind of animals, like look at milk production or egg production in chickens or whatever your commodity is, we haven't seen that with honeybees so much. You take a step forward and a step back just because the mating process is so crazy. So that didn't really answer your question. I think I just threw another.
No, I think it did. It was a bit of a vague question.
Yeah, no, it's and I think we've relied way too much on what's the next chemical to control varroa. So we're finally getting into the point where, okay, let's look at breeding as an answer to this instead of, what we've done to the bees. I think if we had just left those bees alone, like if you take, what's that island? There's an island off of Brazil where they took some California Italian bees and just let them go. And they equalized with varroa. Initially,
I don't think we could have had that amount of loss of bees for
but
our industrial agriculture need for them.
Yeah,
So,
it's the same thing.
couldn't.
But you take huge losses initially, and then it kind of levels out.
It
Or just
does. Yeah.
organic farming, we're starting to move into that because it's just so much more sustainable.
So much more.
Yeah, I mean, it's what we've done in agriculture is really disgusting, actually.
Mhm.
Tom has another question, I think. Tom?
Hello there. Can you hear me there?
Yeah,
Yep.
yeah, go ahead, Tom.
Hi. I'm from northern Vancouver Island, and I received, well, this was a few years back, some Hawaiian queens, and they had to be the most aggressive, mean bees that I ever had in my life.
Oh.
Like, you had to put your bee suit on down the road because they'd meet you at the door of the pickup when you got out. But that being said, I've never had bees that were more productive. Like I've never had bees since that were more productive.
Yeah, I don't think that's related. They had initially, I don't know how far back that goes, but Hawaii was full of Mellifera mellifera. That's the first bee they had there, and it took quite a bit of time to change that over and get more European bees in there. So that there may be some residual of that, but I don't think it's related to. I haven't seen any data that shows that honey production is related to defensiveness.
Okay, because
It's commonly said, though. It's
It
commonly
is,
thought
yeah,
of.
it is commonly said, but I
Yeah.
don't think it's,
No.
yeah, that takes all the pleasure out of it. I just don't have tolerance for that.
well, I enjoyed it because I ended up with a massive crop. But on the other hand, that winter, I couldn't get those bees through the one. I think out of 20 queens, I ended up with 2 that actually wintered over.
Wow, okay.
They just can't handle the moisture. Like I'm from the North Island, they can't handle the moisture up the North
Uh-huh.
Island. But
Yeah, well, they just weren't well adapted. I have no idea what that was. It's hard to say.
oh, that's good. Thank you.
We have another question now from Jerry Rodzima. If you'd like to go ahead.
Yeah, no, I want to comment a little bit more on what Tom said there. I'm also on the North Island. And what we have found is we can have bees in our back lot here. We walk through May and June, and I don't half the time don't need a suit or anything like that. We take them later in the season out to the fireweed, and you need your suit when you're half a mile from the bees already. So here on the North Island later in the season, I think it's a lot more climatic than it is anything else.
Mm.
Well, thank you for that comment, Jerry. Yeah, I think we're right past our time span here, and we don't want to take too much time. But Sue, it's been fabulous. You've really given us a ton of information, and we really appreciate it. And the fact that you're so giving, and we hope to give back to bee research as well here at some point.
Great. Yeah, no, my pleasure. Fun to talk to everybody. It's a good question. Yeah.
Thank you, Sue.
Okay, take care.
Okay. Yeah. Thanks everyone for joining us. And I guess I should suggest that we have another presentation next week, and that'll be a honey producers queen breeding program by Ian Stepler. He's just been going for 5 years, and he's made amazing changes in his own operation, and he's very much self-sustaining. And so I think we'll find that quite interesting as well. Anyway, thank you all for attending, and hope to see you next week.
Yep. Thanks for organizing, Liz.
Yeah, it's all of us.
Thank you for joining us for this presentation. BC Bee Breeders is producing a series of recorded sessions to share knowledge within the scope of breeding honeybee queens. For more information, check out our website at bcbeebreeders.ca. To support ongoing research to be shared with beekeepers across Canada and the world, please click the donate link on our website at bcbeebreeders.ca. Thank you.